Genome-wide association studies, or GWAS, produce a set of genomic loci where there are statistically significant associations between specific sequence variants and the risk of a disease or magnitude of a trait. But the ultimate goal of the research is to identify which genes at these loci are involved in disease risk. Often after performing a GWAS, researchers will combine multiple other data types and methods in order to predict the causal or effector gene at each locus. Usually these predictions and their supporting evidence are presented in supplementary data files, where they are static and relatively inaccessible. We believe these lists are very useful because they give a consensus view of the genes most likely to be important for a disease or trait. So we're curating them from the literature and presenting them in the knowledge portals. To see lists of predicted effectors, go to the Predicted Effector Genes link in the KP Labs upper menu of any knowledge portal. On this page, you can browse all the effector lists or filter the table by multiple criteria. For example, if you're interested in cardiovascular traits, begin typing cardiovascular in the phenotype group filter, select cardiovascular, and now you've narrowed down the table to those lists for cardiovascular traits. The blue View button in the evidence column shows more information about each list, such as whether it was generated manually or by an automated method, or whether it assigns categories or scores to genes. To access a list, click the Go to List button. The top of the effector list page shows the reference for the list and gives a brief summary of how it was generated. Scrolling down, the table of predictions is located on the View Data tab. For some lists, there's also a Research Method tab giving more details about the method. The options above the table allow you to set the number of rows you see, save the table in CSV or JSON format, and show or hide specific columns. You can sort the table by clicking on a column header. Some column headers also display additional information on mouse over. Click the View button to display all the supporting evidence for a row. Above the table, the filters allow you to find results of interest in the table. In this example, if you type a text string into the gene name filter, you'll find all rows where that column contains that text string. Other filters, like the number of evidence filter here, allow you to set an upper or lower threshold for a numerical value. Some filters also have drop-down menus that allow you to choose between options. Finally, for more written documentation, you can go to the tutorial link at the upper right. We're very interested in displaying predicted effector lists for a wide range of diseases and traits. If you've generated a list and would like to display it on the knowledge portals, please contact us.